This lecture is going to be based on what happens during DNA replication when DNA replicates during interphase S of both meiosis and mitosis. When DNA replicates, it's supposed to make an exact copy of itself, and many times it does, and that leads to probably no issues. But sometimes the DNA, when it copies itself to put into new cells, it makes mistakes. And so it actually changes the genetic code of certain genes by doing that. Uh, you can substitute one base for another. And so, in other words, if there's supposed to be a G in a certain spot on the DNA and it copies itself, and instead of putting that guanine or a G in there, it puts a C or a T or another base, and that's called a substitution. It's just one wrong base in the DNA ladder, and it creates one wrong codon. And as you know, codons on the messenger RNA that comes from the DNA code for amino acids. So if you have a wrong codon on the messenger RNA, you could actually put an incorrect or a different amino acid in the protein you're making at the ribosomes. So what happens there is that the protein might not work right. So what happens is that you get either some sort of genetic disorder because that protein is not going to operate properly. Very rarely it actually operates better, but that's another story coming later on. We're going to give you two examples of substitution mutations like that, where just one base is substituted for another on the DNA when it replicates. And those two examples are substitutions that lead to sickle cell anemia and another disease called PKU. A frame shift mutation, that's a different story. That is when you actually leave out a base when the DNA replicates. That's called a deletion. And so that means that the whole sequence of bases on the DNA is moved up one, and that can affect every codon from there down to the end of the gene. And you can put lots of wrong amino acids in the proteins. That can lead to very big problems. An addition would be one that you actually add an extra base into the DNA. And again, that would alter the shift, would change the whole frame of the DNA molecule, one base. And so all the codons will be spelt wrong from there down. And you'll put in, again, wrong amino acids in many places. So these are issues that could happen when DNA replicates and sometimes doesn't do it right. This picture here is actually an amazing photograph of a series of ribosomes. A series of ribosomes is actually called a polysome. And you see many of them here. These are the big black dots. And in these ribosomes, you actually see a string that's going through them like this. You can see them in various places fairly clearly. That string is messenger RNA, and that came from the DNA and has the code, for making the proteins, the three base codons to put the proper amino acids in the protein as the messenger RNA moves through the ribosomes. These little strings that are coming out of the ribosomes are the actual proteins. These are the amino acids that are being linked together by the ribosome to produce whatever protein this picture is showing here. Hopefully this is all working properly. The messenger RNA has the proper codons. The DNA didn't have any errors in it. And so the protein will be made properly. But if, let's say, the messenger RNA had an incorrect codon somewhere around here, when that gets to the ribosome, it's going to put a different amino acid in the protein that it's making. And that could alter the effect of the protein, and that could have a very serious effect on the organism. The first example we'll talk about here is called PKU. PKU is a disease or a disorder called phenylketonuria. We'll just call it PKU. It's causing the amino acid phenylalanine. Phenylalanine is one of the amino acids in proteins. And normally when you eat proteins, the amino acids can be burned up in the mitochondria. That's fine. You can get some energy that way. But if we eat more protein than we need, you have to do things to those amino acids so that they don't accumulate in your blood. So what happens is that you have an enzyme that actually is supposed to break down extra phenylalanine that you are accumulating in your body by eating a lot of protein. Normal people, no problem. They break it down easily, and the phenylalanine does not accumulate in the blood. But if you have PKU, the enzyme that breaks down phenylalanine, which is a protein, is not made properly because of a substitution mutation in the DNA. One base is incorrect. One amino acid, therefore, is put incorrectly into the enzyme that's supposed to break down PKU. So you can't break down you can't break down the phenylalanine. So what happens is that the more protein you eat, the more phenylalanine accumulates in your blood, and it can cause all sorts of problems in your body. For the rest of their lives, people with this have to be careful what they eat, because if they eat a lot of protein, they're eating phenylalanine, which is one of the amino acids in the protein, and that could actually damage them because you can't break the phenylalanine down, and it'll accumulate in your body. Now, if phenylalanine does accumulate, you get neurological problems. The baby's normal when he's born, by the way, doesn't seem any problems, but every child in the United States is tested for PKU when they're born. They take a little drop of blood from the baby, and usually from his heel, and they test it to see if there's elevated phenylalanine in the baby's blood, and he may have PKU, and then they do further tests to see if he does. If he does, you have to give the baby a special diet, so that's very low in phenylalanine, so that he just has enough phenylalanine to make his own proteins from, and burn up a little bit in the mitochondria, and not have any extra, so that it doesn't accumulate in his blood. If you do have PKU, though, and you don't, and you do eat, proteins a lot, and the phenylalanine does accumulate in your body, you could have neurological problems, seizures, skin rashes, normally an abnormally small head that forms in the baby as he grows. You'd be very hyperactive. You can have intellectual problems, developmental problems, many behavioral problems, emotional, social problems, and even psychiatric problems. So you can see this is a very serious disease, but it's one of the few genetic disorders that can be modified by diet. You just have to give him a special diet that's very low in the amino acid phenylalanine. So because he can't break it down, the protein that's supposed to do that for him is not made properly because of the substitution mutation, and he won't have a problem if he doesn't have a lot of phenylalanine in his diet. So for his whole life, he has to eat this special diet that's very low in he can't eat high protein foods for the rest of his life. So that's PKU. Most of the time, the parents don't have PKU. They are carriers, because you remember, you have two genes for everything. And if you look at the diagram here, you see the father has a normal gene, this one, and he has a PKU gene. But because he has the normal gene, he is diploid. He has two genes for every trait. He doesn't have the disease because the normal gene will make the enzyme properly. But he carries the PKU gene also on his other DNA that he got from his other parent. And so he could actually, in meiosis, put that in his sperm. Same thing is true for mom. She can put her bad gene in her eggs. So if a sperm from him has a bad gene in it, and an egg from mom that's a bad gene in it, you can make a baby that has two bad genes. And that's most of the way you get a affected child like that. If you do the genetics problem, it's a 25% chance that two people that are heterozygous, in other words, they have both types of genes in them, can have an affected child, 25% shot. Here's the nice picture of that. You can see here's the DNA. The blue are the introns that are going to be taken out, and the green are the exons that are the good messenger RNA, sequences that are going to be left in. And then inside the nucleus, the cell takes out all the nonsense DNA, the introns, and links together the exons to make the proper messenger RNA. The messenger RNA will go to a ribosome, and when it goes to a ribosome, it'll be read codon by codon, and you'll create the protein that you're supposed to make. This is normal protein. This is the one that the enzyme that breaks down phenylalanine properly. But if you look at the codon here in this exact spot, it's GAA, and that's supposed to be that way. It puts the proper amino acid in this location, and the protein folds up properly and becomes the proper enzyme to break down phenylalanine for the child. But if you look at the PKU gene in this exact spot right here, there is not GAA but AAA. You notice there's one mistake. In other words, instead of being a G there, there's an A there, and that's a different codon. That codes for a different amino acid. At this spot in the enzyme, that's supposed to be GLU or glutamic acid. Here, this is lysine, a different amino acid. And because it's a different amino acid there, this protein folds up differently and doesn't attach to the phenylalanine properly, and so it can't break it down. And therefore, if the kid eats a lot of protein that has a lot of phenylalanine amino acids in it, the phenylalanine will accumulate in his body and cause all those problems that we just talked about. Now, another type of disease that you can get from a substitution mutation is sickle cell anemia. Sickle cell anemia is a disease where the red blood cells here, which is supposed to look like little poker chips, it's supposed to be kind of flat with a dent in the middle. They look like that, pretty bad. Now they still carry hemoglobin and they can still carry oxygen, but the shape is really, really different. And so it has trouble getting through, or the capillaries, and causes clogs and big problems in your body. This is caused by one substitution. And the sixth amino acid in the protein hemoglobin, which is the red protein inside these cells, is the hemoglobin that carries oxygen for you. The sixth amino acid in that chain of hemoglobin is a protein, is substituted. Instead of glutamic acid, it's valine is put in there by mistake. And because of that one spot on the hemoglobin chain, the hemoglobin causes this effect on the cells. It's very serious. If you look at this picture here, you see where there is a reason why sickle cell anemia became fairly common in dark-skinned people that come from Africa. You can have people that have a very high percentage of them actually contain this bad gene. And it could be as many as 1 out of every 400 African-American births would carry this bad gene. And that's a lot of people. And let's see why that a bad gene actually became that common. Usually that doesn't happen. Usually if you have a bad gene, it kills people and they don't live long enough to have babies. And so the bad gene kind of disappears in the population. But here, the gene became much more common. And if you look at the dots, this is an area where there's a lot of malaria. Now, malaria is a very deadly disease, probably caused more deaths than anything in history. And it's caused by a mosquito bite. And what happens is that if you have sickle cell anemia, the organisms, the little protozoa that's in the mosquito saliva that gets into you, actually has to attack your red blood cells. So if you have the gene for sickle cell anemia, the mutated gene, the substitution, if you have 2 of them, if you got it from both parents, then you're probably not going to live very long, because your cells will sickle and have trouble getting around your body and cause all sorts of problems, while you're still young. But if you have one good gene and one bad gene, it turns out that you don't have sickle cell anemia. The cells will maintain their proper shape. You'll carry the bad gene that you can give it to your child, but you don't have the disease because you have one good gene, one good gene, one bad gene, because of the 2 parents. But if you have what they call sickle cell trait, which is the heterozygous condition, one good, one bad, you are basically immune, or very close to be immune to malaria. And that's actually an advantage. So here you have a bad gene that doesn't make you sick because you only have one copy of it. You can give it to your child, but it gives you an advantage to live longer in these areas of the world, because you won't die of malaria before you have children. So that means that people who have that sickle cell gene, only one copy of it instead of 2, will have an advantage, and the gene will, they'll be able to make babies pretty easily because they'll live longer, and so that gene will become more common in the population. So here you have an example of where a substitution mutation, which is just one base substituted for another in the messenger RNA, puts one wrong amino acid in the hemoglobin protein. And as long as you have only one copy of that bad gene, and you live in an area where there's a lot of malaria, and there's no, obviously back then in olden days, there wasn't any cure for it, you would actually have an advantage and be able to live longer and make babies more frequently, and therefore carry on that gene and make the gene become more common in the population. That's why that almost all the people who have sickle cell anemia, even in the United States, come from Africa. Now, if you do have it, these are the normal cells. That's what they're supposed to look like. If you have sickle cell anemia both times, in other words, you get it from both parents and you're homozygous for it, you have bad gene and bad gene, all the hemoglobin you make in your red blood cells is going to be the wrong kind, and it'll have that mistake in it. And these are all the things that will happen to you. So if you look at the picture here, you can see all sorts of breakdown of red blood cells, clumping, clogging of small blood vessels, accumulation in the spleen, all these other things, pain, fever, brain damage, spleen damage, anemia, mental health problems, pneumonia, kidney failure. So you see, these poor kids don't live too long. Very, very serious disease. But if you only have it once, in other words, you have one gene that's good and one gene that isn't good, and inside of your cells, the good gene will make the hemoglobin okay, and your cells will be looking the way they're supposed to, but you can still carry the bad gene. And of course, when you do meiosis, you put them into sperm or eggs, and so you'll have a sperm or an egg that actually has a bad gene in it that you can give to your child. Now, if mom also is heterozygous and she and her egg has a bad gene in it as well, and they meet, then the kid will have bad, and that'll be homozygous bad, and that kid will have sickle cell anemia. Again, that would be a 25% chance of two homozygous people that don't have any symptoms. I mean, heterozygous people, excuse me, that don't have any symptoms can have a baby that does. Because they have that ability to make eggs and sperm, which have the bad gene in it. And if they have a bad gene from a sperm and a bad gene from an egg meet, which is a 25% chance if you do the Punnett square, you'll see a baby that has sickle cell anemia. It's a very interesting disease. It's caused by one mistake, one amino acid wrong in the hemoglobin protein inside your red blood cells. It's just a substitution. Now, these errors here, these are chromosome errors. Now, chromosome errors mean that there's changes in the actual chromosomes during meiosis or mitosis, mostly meiosis. And it causes much more serious problems because it changes more than one amino acid in the proteins. These are frame shift mutations. They leave a protein out, or they leave a base out on the DNA or put an extra base in, and that changes all the codons from there down to the end of the gene. Now, if you have a deletion, an example I'll give you of a deletion protein is called cry of the cat syndrome. Cry of the cat syndrome means that the kid will, when he's born, he has a larynx or a voice box that's not developed properly. And because of that, when he cries, he sounds like a crying cat, very high pitched cat sound. So they called it cry of the cat syndrome. But that's not the only problem he'll have, though. He'll have, serious organ defects, kidney problems, and all sorts of life-threatening complications that usually result in death. Most of the fatal complications that lead to death occur before the kid is even 1 year old. Now, that's caused by a deletion in the gene. One amino acid is left out of a protein. I'm sorry, a base is left out on the messenger RNA, and that causes the whole strand of messenger RNA to move over one base, and that basically changes the spelling of every single codon all the way to the end of the gene, and that causes proteins that are really messed up. And that causes, one example of that would be the cry of the cat syndrome. Duplication is when the DNA replicates And then what happens is during crossing over in meiosis in prophase one, uh, a piece of one chromosome breaks off and is supposed to cross over to the other side on the homologous chromosome and just exchange pieces like they do. But when it goes over there to the other chromosome, the copy on that chromosome doesn't come off. And so what happens is you have the gene duplicated. You have the gene is missing on one chromosome because it came off of it to try and cross over to the other one, but the one on the other side just stayed there. So you have that gene there twice, and that could happen many different times, and it has happened in our times, in the past many different times. We call that multi-genes. We actually have the gene repeated over and over again on the chromosome because of mistakes in crossing over. And what happens is that now, if you have, let's say, for example, genes for skin color, if you have, let's say, 8 genes for skin color repeated over time, over the many generations of people that came before you, and they made this mistake in doing crossovers, and they kept duplicating that gene, you might have 8 or 9 genes on there that have to do with making skin color. If they're all dominant, that means your skin color is going to be pretty dark, and those would be people who have black skin. But if half of them are recessive and half are dominant, then you'll have lighter skin because the recessive genes don't make the melanin. And then, you might have only 2 or 3 of them working, and then you'll have even lighter skin. So this explains a little bit about why there's so much variation in things like skin color and eye color and hair color. It's because of these genes that have accumulated on the chromosome to make the same protein over and over again. It just depends how many of them are dominant and how many are recessive, and in any one person. And that way you can make different variability, have different variabilities in the effect, skin color, hair color, whatever it is. An example here, though, is an interesting one. The growth hormone gene and the prolactin gene are in the same family. They're very, very similar. And it turns out that we think that the gene for growth hormone, which is a protein that you make in your pituitary gland that makes you grow, and the gene for prolactin, which is the hormone that actually causes women to make milk in their breasts when they have children, are very much the same. And so it turns out that we think that during our genetic history, there was a mistake in crossing over during prophase 1 of meiosis in somebody, and the gene was copied twice on the DNA that received the piece of DNA and didn't give one back, and so you have the gene there twice. And then one of them mutated just a little bit, probably a substitution mutation or something like that, to become prolactin. So now you have 2 genes, one to make growth hormone and one to make prolactin on the chromosome. And that allowed mammals to actually not only grow, but make milk for their babies. So that could have been caused by an actual duplication. You make 2 genes on the chromosome that initially did the same thing, and have one gene slightly change over time because of maybe a substitution error or a series of substitution errors, to be able to make an altered protein, and which would be very similar to the growth hormone protein, but have a different effect on the body, and that effect would be making milk in the breast. So those are duplication errors. Inversions are very serious. That's when a piece of DNA is supposed to cross over to the homologous one on the other side, and from the other parent during meiosis prophase 1. But when it gets over there, it flips over, and it's like reading the codons backwards. So you might have a whole series of codons upside down on the chromosome that receives the piece, and so you read, every single one of them is read wrong. And so you put lots and lots of amino acids incorrect in various proteins that you're making there. And a lot of times that leads to different types of cancers. A translocation is an interesting one. What happens there is that a piece of DNA can come off of one chromosome and it's supposed to be, crossed over to the homologous one over there from the other parent. But it misses, it doesn't stick on. And so it floats around in the nucleus as a little piece of DNA that broke off the chromosome. It's supposed to cross over, and land on the homologous chromosome, but it did not. And so it lands on a different chromosome, and it's translocated onto a different chromosome. And so that can lead to other cancers like leukemia. Now, these are how they actually work. You see, this is the normal DNA here, and you see that piece here, it's missing on this one. And so all the codons on this DNA is going to be from that point down is going to be incorrect, and you're going to make altered proteins, which may or may not work right. That's a deletion. You leave out a base on the DNA. A duplication. See, here's two genes here, or two bases, and they're supposed to cross over to the homologous chromosome on the other side during prophase 1 of meiosis. And when they do that, though, the genes from the other side stay there, and so you end up having it there twice. And that's why we have multi-genes, genes that are repeated on the chromosome over and over, and you could have different things like different hair colors and eye colors, and which ones are dominant, which ones are recessive. If they're all dominant, you'll have dark hair. If only half of them are dominant, you'd have lighter hair, and so on. This one here is an inversion, and you can see that the gene of BCD was supposed to cross over to the other side on the homologous chromosome when they're synapse during prophase 1, but it flipped over, and now it's DCB, and that causes a problem too. A lot of cancers are caused by that. And the translocation means that the piece of DNA is supposed to go onto its homologous chromosome, but it goes onto a different chromosome. And again, that can cause also problems. This is an actual photograph of this little piece of DNA here that's been translocated onto a different chromosome. It's supposed to be on another chromosome, but it crossed over incorrectly, and it went hooked onto that green one instead of the red one it's supposed to go on. See, this was supposed to go on to here, like that, but instead it went on to the green one, which you notice it doesn't have a piece of red on it. And so this is a translocated gene, and so the genes on this chromosome are going to be read wrong, and make proteins that are modified. Most of the time will not work properly, and that also leads to many cancers. Now, another very interesting one is called non-disjunction. Non-disjunction is what happens when, in anaphase, the chromosomes are supposed to separate, and they're supposed to go down the spindle and end up in two different cells. Well, sometimes they don't separate and they go down together. Non-disjunction means they don't separate, so they go down together. Sometimes it's called aneuploidy. And one of the most common genetic disorders in humans in the whole world is caused by this, and it's called Down syndrome, where chromosome 21, the 21st chromosome we have in our set of chromosomes, when they line up on the spindle in meiosis metaphase 1, they're supposed to separate during anaphase 1 and go down the spindle on either side. But what happens is, of course, that they don't. They stay together and they go down as a team on one side and nothing on the other side. And so that means one egg or one sperm that are being created by this meiosis is going to have an extra 21 in it. And so then when that egg is made, it'll have two 21s. If it's fertilized by a sperm that has 121, then you get a kid that has 3 21 chromosomes instead of the normal 2. And that's what causes all the Down syndrome symptoms. It's an extra chromosome 21. It's just caused by that process, non-disjunction, where the chromosomes do not separate properly when they're supposed to move down the spindles. And you end up with extra chromosomes in the egg or the sperm when they're finished. Klinefelter syndrome and Turner syndrome, they occur on chromosome 23, which is our sex chromosome. And you can see that these two X's were supposed to separate during meiosis, but they didn't. So they went down together, down the spindle, and ended up in an egg eventually. And that egg had two X's in it instead of the one X it's supposed to have. And of course, it got a Y from daddy, because a Y sperm fertilized it, because half of daddy's sperm are Y and half of them are X. If it fertilized by a Y sperm, then you'll have an XXY, and that's Klinefelter syndrome. Now, Klinefelter syndrome is kind of interesting because it's symptom that is caused by having that double X chromosome in a boy. Now these guys are all boys because they are it has the Y chromosome. You could have as many X's as you want. If you have one Y, that makes you a boy. And so what happens to these poor kids when they get born is they start to grow up. They could have a very low sperm count. They would not be able to make babies. Very, very low or no sperm at all. They have very small penis, very small testicles, very low sex drive. They're very tall. They have weak bones. They have decreased facial hair and body hair. They have enlarged breasts, increased belly fat. So in a way, they're kind of sort of feminine in a few different aspects like that. But they're not going to. They're also sterile. This one here, XO, means that the egg didn't have any X in it. It's the one that didn't get the X. Remember, they're supposed to go down. One X supposed to go one way, and the other X supposed to go the other way during meiosis. But if they go together, the other egg that's supposed to come from the other side gets no X at all, and that would be the 0. And if that's fertilized by an X sperm instead of a Y sperm, you get Turner syndrome, which is basically a girl. There's no Y chromosome, and just basically a normal girl. She's normally a little taller than normal, a little slimmer, but not too bad off. That's called Turner syndrome. If that was a Y sperm, by the way, it would never grow because the Y sperm is missing many genes, and it would not an X Y without an X would not make a baby properly. You could also have 3 X's, an XYY. There's also all sorts of different ways you can cause the genes to be different because they don't separate properly in meiosis. Okay, and here's just an example of that. You see here, these chromosomes here are supposed to separate during anaphase. They're supposed to go down there as double chromosomes because this is anaphase 1. And but you see these two guys, they're going down there together and they're not separating. That's going to create an extra chromosome in the egg or the sperm that's being produced here. Okay, and a missing one in this one. See that whole big chromosome is missing in these guys. That could be a problem. Over here on the other side, you could also have a mistaken meiosis too, where the chromosome is supposed to separate. This one's supposed to go this way, and that one's supposed to go that way. The centromere is supposed to split, and they go down together. Go down on either side, but these two guys are not splitting, and they go down together, and it has basically the same effect. You end up with 2 big chromosomes in these cells, and no little chromosome, only 1 little chromosome in this one. It's really an issue that can that causes so that extra chromosome can cause all sorts of problems. And this one, of course, is missing a whole chromosome. So if that's an egg that actually gets fertilized, it could be a problem. If this is an egg that gets fertilized, it could be a problem. That's non-disjunction. Now, this is a picture of a Down syndrome child. These are here the Down syndrome's chromosomes. You have 2 chromosome 1s, they look normal. 2 chromosome 2s, 2 chromosome 3s, and you keep going all the way to chromosome 23, which is the sex chromosome here. This looks like there are 2 X's, so that makes this a girl. But look at chromosome 21. There's 3 of them. That little difference, that extra chromosome here, creates all the Down syndrome symptoms. And we know what that is. Down syndrome children can have, short attention spans, upward slanting eyes. They have wide, short hands, little thick fingers. They have developmental problems. They have impulsiveness. They don't learn very quickly. Delayed language, delayed speech. They have a tongue that's very thick and sometimes hangs out of their mouth. All of that is caused by an extra chromosome 21. And, their lifespan is also shorter than the average person. This one here is Klinefelter syndrome. This person has 3 sex chromosomes. Chromosome 23, 1 Y that came from his father, and then the two X's. That means that in the egg, there were two X chromosomes. They did not separate properly in meiosis, and so the egg ended up with two X chromosomes, and when it was fertilized by a Y sperm, it became this XXY, and that's the Klinefelter's one that has all those symptoms we talked about a few minutes ago. Okay, and this is an XYY one. You see it has two Ys. Now, if you have two Ys, it's fairly normal boy, but you could also have some issues with behavior and some other things that could happen to you if you have two Y chromosomes. It's not that uncommon. A lot of people believe that it creates a little more aggressiveness in the child, more likely to get into fights and so on. But otherwise, it's just an extra Y chromosome doesn't have that much of an effect on you. It's XYY. The XYY people normally, though, are a little taller than average. They are, if you look at their pinky finger, it's kind of interesting. It's curved. You can tell an XY, one of the ways you can tell an XYY individual is by looking at their pinky. Instead of being nice and straight, it has a very marked curve in it. And they have widely spaced eyes. And when they're XYY, when they're teenagers, they have a lot of extra acne that happens, unfortunately, on their faces. But they do live normal lives. Now, let's take a look at this, and we'll read it and know how important it is. It says the errors that occur during DNA replication, meiosis and mitosis, are called mutations. These errors. Now, you see, in what we talked about so far, most of the time they lead to something bad. It's easy to make something not work right. The vast majority of the time, they result in a protein that does not work properly. Rarely, they can change a protein, however, for the better, altering the phenotype in such a way as to give its bearer a reproductive advantage. In other words, if you're an animal and you are, let's say, have a light color fur, and there was a mutation in your DNA, a substitution mutation or a frame shift mutation or some sort of a addition or deletion of a base on the DNA when the DNA replicated, and it created a protein that's a little different than for the color of your fur that it was supposed to be, and you end up being a little darker as a child because you're born with that mutation from your parents. And that may make you actually blend into your environment a little better, harder to see you. And if you live in an ecosystem where you are the prey, boy, what an advantage that would be for you. And if you have an advantage like that, your chances to live longer and make more babies than those that don't have that advantage, over time, you're going to have more animals that look like you as the generations go by, and then that look like the older parents that had lighter fur. So this, along with the endless rearrangement of genes within sexually reproducing populations, results in slow changes in species. In other words, they change to, and any change that gives them any kinds of advantage or success in their very difficult environment to live in. We live in a nice, safe world. Of course, there's things that happen to us too, but nothing like if you were living in an actual ecosystem where things were trying to eat you every day. And so if you have any advantage, boy, that would give you a big hit. And so what happens is that, you can make more babies than those that don't have that advantage. And over time, the species will end up being more, the members of the species over many generations will end up looking like you. And that's change. And that's change that makes the organisms like more suited to their environment. And the environment kind of picked out those that are better suited for it. That change is called evolution. And that's going to be our next topic this week. All right, so this was our lesson for the first part of the week. And then on the middle of the week, you'll have another lecture on evolution. And that's it for this week as far as the lecture goes. I hope that helped you.